Hello everyone, it's my pleasure to introduce our paper, Agriculture Increases Potential Health Risks of Vertebrate Viruses in Soils. Vertebrate viruses could adversely affect the health of human and animals, and trigger broader socioeconomic consequences directly and indirectly. The increasing frequency of pandemics of vertebrate viruses causes severe global distress, and increases common attention on One Health, which stresses the roles of interconnections between hosts and their shared environments in achieving optimal health outcomes. Soil is the foundation base of terrestrial organisms, and the critical interface of their interactions. It is regarded as a cornerstone of One Health. Although soils have the most diverse and complex microbiome on Earth, the occurrence of vertebrate viruses in soils is still unknown. Also, it's critical to assess the effects of agriculture on the prevalence and health risks of vertebrate viruses in soils, since agriculture is one of the most important mediators of pandemics. Highlights: First, agricultural soils have higher detection rates of vertebrate viruses than natural soils. Second, human-related viruses dominate abundance, but not diversity in agricultural soils. Third, most soil vertebrate viruses are conditionally rare with stochastic distributions. Last, agriculture increases potential health risks of vertebrate viruses in soils. To explore the occurrence of vertebrate viruses in soils, we mapped against a manually curated database of vertebrate viruses with 3,000 soil metagenome and 1,400 soil metatranscriptome sequencing data across the globe, spanning a wide range of terrestrial ecosystems. Totally, 128 and 134 vertebrate viruses were detected from nearly 40% metagenome and metatranscriptome samples, respectively. Although human-related viruses were detected in less than half of richness, they occupied more than 75% of abundance, and their prevalence patterns across soil samples had strong ecosystem specificity. The detection rate in agricultural soils was much higher than in natural soils. Only 30% of vertebrate viruses were shared between agricultural and natural soils, in both metagenome and metatranscriptome samples. Their occupancy had a significantly positive correlation with abundance. This linear relationship represents a gradient ranging from a restricted spatial distribution with low abundance to wide distribution with high abundance, leading to the concept of conditionally rare taxa that the bottom left species have the potential to bloom. This gradient can be caused by stochastic processes, which brings in the challenges of preventing or mitigating vertebrate virus pandemics. We further link their average abundances with spatial variances, which were well described by Taylor's law, implying a dependency between average and variance of abundance across soil samples. The average abundance alone could characterize the distribution of abundance fluctuations of each vertebrate virus in soils. Combined with the properties of conditionally rare taxa, it is vital to apply offensive strategies in high abundance areas of high average abundance vertebrate viruses in soils to limit their dispersal, while employing defensive efforts to reduce the vulnerability of low abundance areas to colonization. We also tested the abundance richness log-log relationships to reveal coexistence patterns and mechanisms of vertebrate viruses in certain ecosystems. Remarkably, the richness in agricultural soils possessed weaker linear correlations with abundance than in natural soils. Instead, their richness in agricultural soils formed a hump-shaped relationship. The most popular explanation for this relationship is the intermediate disturbance hypothesis. It postulates that diversity peaks at an intermediate level of abundance and disturbance, whereas at low abundance or high disturbance only a few opportunistic species could exist, and at high abundance or low disturbance only a few top competitors persist. The low richness and high abundance conditions in agricultural soils may be caused by anthropogenic disturbance, and few species in the local pool adapted to these conditions. Given that most vertebrate viruses might colonize and extinct stochastically, vertebrate viruses would immigrate from distant pools and could simply replace the old ones by increasing abundance, complicating the coexistence of viruses in soils. Finally, we assessed the risks of each soil sample. As expected, agricultural soils posed the highest risk of vertebrate viruses in both metagenome and metatranscriptome samples. Further, we calculated the correlations between the values of risk index with environmental and anthropogenic factors to identify potential drivers of sample risks. Overall, the values of risk index were significantly positively correlated with precipitation, temperature, human development index and population density, while were significantly negatively correlated with soil fertility properties. Those results indicate that the health risks of vertebrate viruses would be aggravated by the broad array of global changes, such as drought, global warming, and soil erosion, which could be strongly affected by agricultural activities directly and indirectly. In conclusion, soils are an important mediator of vertebrate viruses, and most of vertebrate viruses were conditionally rare in gain or loss in soil samples stochastically, bringing challenges to vertebrate virus surveillance. Further, health risks of vertebrate viruses are aggravated by agriculture-related activities.